beta peptide, and rate of its expulsion is higher in a healthy brain. What happens? That either aging or some metabolic or immunological diseases, the production rate is more than its excretion rate in the brain. So that's why it is depositing in different areas of the brain. Amyloid itself is toxic. So therefore, we must reduce the amyloid burden on the brain, and there are different reviews, even PNS and all good journals, recently 2018 and 2019. So the point is that our drug development strategy should be modified, not the target. And amyloid beta peptide and also p-tau is the byproduct of amyloid beta peptide deposition is correct, but we need certain strategies to target this amyloid beta peptide to have benefit of treatment. But it is not right to say that amyloid beta peptide is wrong target. In our opinion, at the moment, in the line of work. Another question? Yeah. Yes? Deliver the drug, yes.
And then after it's destroyed the data and all those like the particles things after it's destroyed.
Yes. The question is, what will happen or what is the fate of nanoparticles or nanowires when they are delivering or complete their work? The answer is, nanoparticles normally do not leave the brain tissues or the body for a longer time. They can be accumulated either extracellular space or within the cells. Therefore, this is very important to choose the nanoparticle or nanowires that are most, not harming or innocuous. We are so far using with Dr. Tian this titanium dioxide nanowire. They are innocuous at least for several years. And even in Arkansas and Little Rock, I have this knowledge that when they are doing knee operation, they fill with this titanium wire deposits inside the knee and close the operation, so the knee is healing faster and long time. And even after 6 years, there is no complaint by the patient of any kind. So we believe that we have to choose innocuous material because they can live inside the body for longer period. So far, that's our knowledge. Any other question? Yes, please.
So to what extent do you think this nanowire will be safe to be injected in the body? Because it's a spike, like a spike in the oxygen, and you might have a concern about this.
Yes, as I showed some of the examples that these nanowires, if given intravenously, they can pierce the endothelial cells, and very tiny hole, less than 0.1 micron or something like that, and that nature can repair itself without showing any damage. Our experimental limitation is up to 6 months. Up to 6 months, even we just inject nanowires, either intravenously, intraperitoneally, even inside the brain or over the spinal cord. We don't see nanowire induced any pathology up to 6 months. That's the permission we have. And considering the clinical trials in Arkansas, we believe that 6 years human knee can have, at least they have the data, not showing any pathological effect. So the titanium innocuous nanowire can survive within the body for a longer time, but we need to understand its physiological properties for a longer time and dosage.
I have a question that for a long time that can I get a chance to ask since you are here at but we have Tyson Foods nearby.
Yes.
That's the world's largest meat producer or protein producer, but they have so many nationwide slaughterhouses and slaughtering probably millions of pigs every day. And then how likely that we can kick around, try to tell the Tyson Foods that we can team up with them and harvest
Yeah.
the cellular processing from the slaughterhouses that are otherwise wasted? Can we further develop it into the larger scale manufacturing of the production of the drug that are already in the market or in test in Europe and in China? How likely we can do that? Because we have a lot of people, young guys, all have the interest.
First of all, I'm not qualified to answer this question. And according to my knowledge, working with Ever Europharma company, of course, these are licensed, they are produced from pig brain. And early 20 years ago, there were lots of questions that how it can harm the human body. But there are several, and it was tested in China, it was tested in Hong Kong, it was tested also in the United States, and several clinical trials in stroke, traumatic brain injury, and Alzheimer and Parkinson. They found that a certain dose that is given to a human is totally innocuous, do not provide any side effects up to a certain period, so it is quite safe, and these papers can be found in the literature. But no single person or laboratory, of course, can produce this highly purified peptides or a combination of them, because I guess that to produce 1 milliliter of cerebrolizin, it requires, I may not be correct, it is just a wild guess, at least more than 10 or 15 kg of the brain. So I mean, and it is, they have commercial properties, commercial manufacturing. So not easily can be produced, otherwise many countries or many organizations have produced this kind of drug. So still so far we have this Ever Europharma production, and they are doing it meticulously, and the quality control is very important, and that's why I believe that this is not a one-person job. So use commercially produced material that is qualified, purified, and used for human use. That is, but I'm not qualified to answer your question.
But then in the mixture of the cellular processing, which it's a mixture of the peptides of the different types, that which one or which ones might be more useful than the other, that if that's the case, we can identify them. We probably can inspire biochemistry labs to produce that, mass produce it. I mean, to synthesize it in the lab. Is that doable?
The analysis of cerebrolizin is already available in public domain, and can be found what is its component, how much percentage of each component is there. But still I believe that a large manufacturing company can produce. Likewise, Siemens produces computer, United States produces Boeing, and everything is known, but all countries can produce. Even they have the capacity, they have the money, they have the laboratory. So that's why we have the trademark. We trust, for example, the brand. And that's why I should say that Ever Pharma is producing cerebrolizin. They are very pure, scientifically tested, lots of manpower and lots of money is involved in that. And if one would try to do that, theoretically one can do that, but the quality and the, its purity may not be the same. Even one manufacturer have different batches, and the product can be different. Is that right? I can only answer to you only this. I have no qualification to answer the question.
So
I
in
think it is like
in the sense that we can, in theory, we can team up with these companies that to further develop, right?
I believe so. That's why we have this national innovation. 3 times we have won the award on cerebrolizin, and many American companies want to have patent. The company refused because we don't want to go ahead, even Department of Defense and all other organizations, because something is lying with the company. They do not want to share with that. You can use the drug, buy the drug, do the experiments, and be happy. That's what I am told. But beyond that, I don't know.
Okay. Any other questions?
Yeah. Yes?
So when you verify the cerebrolizin through the lab, and then it destroy the milliliter,
Destroyed or metabolized quickly, I don't know. But the number of amyloid beta peptide deposits are less. I don't know this is because of destruction or because of this, it's excretion from the brain or something like that.
But you come like a smaller,
We are using immunohistological techniques. We can identify whether an beta peptide is there or not, and the numbers are there or not, whether they are becoming smaller or larger, or I cannot say the technique that I am using.
Right, well, okay, go ahead.
Have you observed any therapeutic effect for these nanofiber products, nano specific for the titanium dioxide nanofibers by itself?
The question is very right. We have done in 2007 our first paper of New York Academy of Science, and we have always used the control as the nanowire alone. In our hand, nanowires alone do not produce any pathological changes or behavioral alteration. We have published one paper in one of the brain research by my wife and me, only injecting nanoparticles, not nanowires, and studying different doses and timing. And at certain toxicological doses, of course, they can have some behavioral changes and some minor pathological changes. But nanowires, even in the doses used, even 5 times more than the pharmacological doses, we don't see any pathological changes in the brain or behavioral alteration. That's only I can tell you.
Any other questions? Well, not we can certainly communicate with Professor Sharma if any interested in developing collaboration. Email him. So let's thank Professor Sharma again.
I thank you all for being here and listening to this talk and giving your comments. Thank you for being here.